Yes. Good morning and good afternoon, ladies and gentlemen. It is a great pleasure to have you all today for the 4th edition of Boston-Paris Biotechnology Summit. This is a bridge to collaboration on COVID-19 from both sides of the Atlantic. Please launch the first slide, please. Please, Ethan, please launch the first slide. Okay, I am Soheila Garakanian, co-founder of the Boston Biotechnology Summit and president of Blend of Concept, the summit producer. First, I would like to express our gratitude to the outstanding speakers and moderators for their presence today. Their engagement, know-how, and expertise regarding this pandemic and its societal and management issues are priceless, cherished, and reassuring for all. Next slide. Since 2017, the summit is supported by prominent companies and organizations. We are thankful to our generous sponsors, McDermott Will & Emery, PPD Biotech, and CPL Physicians. The majority of them have been present from the first edition. 4 years of collaboration, trust, support, and excellent relationship mean a lot to us. Next slide, next please. Today, the majority of the audience is from USA and France, but this is really a global meeting as you see it from the flags. Thank you for your presence. End of slides. I would also thank the Mass BioHub team and Midnight Brunch for managing the technical Zoom platform today. Now I turn over to Dr. Praveen Chaturvedi, a leader in life sciences ecosystem in Boston, CEO of Oceanics Pharmaceuticals and our honorary chair. Thank you, Praveen, for supporting the summit since 2017. The floor is yours.
Thank you, Soheila. Good morning, good afternoon, and good evening to all our colleagues around the world. I want to thank and congratulate the co-founders and the organizing team of Shaheen and Soheila Garakanian for putting together the 4th Boston Biotechnology Summit. In addition, I also want to thank and congratulate the International Program Committee that is comprised of many distinguished folks that have helped put together today's event. There is an old adage that states that God always forgives, humans often forgive, but nature never forgives. So here we are in 2020, exactly nearly 100 years after the 1919 flu epidemic, dealing with another respiratory virus pandemic. When one reviews the history of medicine, respiratory diseases as a class are not have been or have ever been under control. Although they are the most frequent cause of death, we do not know how to prevent respiratory diseases and deal with respiratory viruses. It is important to note that respiratory viruses show no correlation between viral load changes and clinical symptoms. Neither are the clinical symptoms predictable. In addition to the challenges of not having adequate control of respiratory viral infections, we need to remember that public indifference, lack of knowledge of the contagion effect of respiratory viruses, as well as the inconsistent periods of incubation of respiratory viruses result in different infectious sequelae. We've all witnessed numerous strains of influenza virus epidemics over our lifetimes, and we've had limited success at best in preventing contagion effect of flu epidemics. So today we've assembled a group of experts that will provide some of their insights and knowledge about what we know of COVID-19 and what progress we have made in our ability to detect and diagnose accurately COVID-19, as well as the success achieved with various prevention measures. These prevention measures include, but are not limited to, social isolation, social distancing, vaccine initiatives, and the panelists will also discuss the progress made towards development of novel antiviral treatments to allow better clinical outcomes for those that are already infected with COVID and or are hospitalized with severe infection. As I mentioned earlier, historically, we've not done well with respiratory viruses and the clinical sequelae. So we expect that we may not have novel solutions and answers to all the questions that we have for the prevention and or treatment of COVID. But I hope that today's event will actually pose the right questions. In that regard, we've assembled 3 panels that will discuss the progress made with our vaccine initiatives, as well as novel therapeutic and treatment initiatives. Since all of this requires capitalization, we will also discuss funding environment to address the global needs. Our creative friends in Hollywood about 25 years ago made a summer blockbuster in which our planet Earth was taken hostage by aliens that intended to destroy the planet. In that movie, it needed all of humanity to come together, and COVID has reminded us that this is a global pandemic that requires all hands on deck. So with that thought in mind, I will turn it back over to Sohaila so that the panel discussions will start asking the right questions. Hopefully such questions as what are we not doing, or what we ought to be doing, or why do we all get it wrong will be discussed today. My sincere thanks to all of the panelists, the moderators, and especially all of you participants from around the world for joining us in this discussion today. Many thanks. Back to you, Sohaila.
Thank you so much, Praveen. Please launch the video, please.
I'm Dr. Shaheen Garakanian, co-founder and program chair for the annual Boston Biotechnology Summit. After 2 successful editions, the first one at the Institut Pasteur in Paris, France, and the second one at the Boston Exchange Center in Boston, Massachusetts, we are now at the Wistar Institute, a world-class research center in Philadelphia, Pennsylvania.
We work together collectively. So collaboration is in our DNA.
My passion is to help patients. Helping patients can be done in many ways. As a physician, treating patients, but also bringing together people, investors, pharmaceutical companies, biotech companies, researchers, and patient advocates together to solve medical problems.
And so think about the 5 things that one has. One has data. From data comes information. From information comes understanding. And from understanding comes knowledge. And from the use of knowledge comes wisdom.
Good morning, good evening, and good afternoon. We would have loved to see you in person, meet and greet you as in a institutional setting, as you saw on the video. Life decided otherwise, and here we are together thanks to technology. My name is Shaheen Garakanian. I am the co-founder with Sohaila and the program chair of the Boston Biotech Summit. I will be co-moderating this session with Brian Malkin from McDermott, Will, and Emery. Brian is an expert in FDA practice and also IP, and is based in the Washington, D.C. offices of McDermott, Will, and Emery. Since January of this year, I have been working hard, like many others, to try to bring novel treatments and preventive agents for COVID-19, setting up clinical trials in Europe, the United States, and South America. It has been very hard. But let me talk about folks for whom the COVID-19 crisis is even harder. First, next slide, please. The Wall Street Journal in August of this year wrote that in a K-shaped recovery, the situation of American workers will be worst, and it will divide American workers. Next slide. The newspaper Le Monde on October 6 wrote that the health crisis has pushed 1 million French women and men into poverty. Next, please. Here in Boston, our main industry is education, and our organizations, our educational organizations, are really struggling with the practicalities of COVID-19. Next slide. Finally, the very serious Economist recently wrote and asked, why are so many governments getting it wrong? This edition has a lot of data and is very interesting to read. Next, please. One of the first medical meetings I attended was in the presence of the head of China CDC. I was impressed by how much he emphasized the need for universal masking. This has now become a societal issue, a philosophical issue, and one of civil liberties. I could not find a better quote than from Albert Camus in The Plague, La Peste, and I will leave you contemplate its meaning. Next slide, please. We will now start our first panel on prevention and vaccination. We are approaching this very strategically, and I will turn it over to Brian Malkin. Brian, the floor is yours.
Hi, sorry, I'm an attorney at McDermott, Will, and Emery and the Washington, D.C. office. I'm a food and drug attorney and in the Healthcare Industry Advisory Group. And it's my pleasure to introduce Dr. Bruce Walker. You can read more of his full bio in the summit booklet, but as a brief introduction, he's a director of the Ragon Institute of MGH, MIT, and Harvard. He is the co-leader of the Massachusetts Consortium for Pathogen Readiness. He is the investigator of the Howard Hughes Medical Institute. He is a Philip T. and Susan M. Ragon Professor of Medicine at the Harvard Medical School. He is professor of the practice IMES and Department of Biology at MIT, and a research associate at the Center for AIDS Program of Research in South Africa. And I'm looking very forward to hearing his presentation today, and welcome, Dr. Walker.
Thanks very much, Brian. And if I could have the first slide. I see Brian, but I don't see the slides. So I don't see the slides. I have a blank screen here. It says I'm viewing.
Oh, the slides are up, Dr. Walker. You can proceed. you
Uh,
can see.
Yeah, but I don't know. I don't know. I can't see the slides, so it's hard to talk from them. Um, they're not coming through on my feed. Should I go to the YouTube channel or what? How do I see your face? Yeah, that in my entire screen. You look quite good.
Can you try view option side by side mode? Will that on the top?
Ah, okay, now I got it. Great, thanks very much. So thank you, Shaheen, and thanks to the organizers for inviting me. I'd like to put this into a personal perspective. Next slide. I finished medical school in 1980, went to Mass General to become an intern in medicine, and thought I was at a place where everybody knew every disease. While I was there, 6 months into my internship, a patient came in with what we thought was just worsened tuberculosis. It was already unusual that a 19-year-old had tuberculosis in Massachusetts. And our presumptive diagnosis was that this was just worse TB. But we got the surprise of our lives when we started working him up and realized that he had two diseases, pneumocystis carinii pneumonia and Kaposi sarcoma, that I had never heard of in this kind of patient population. And on top of that, he also had a B-cell lymphoma in his brain. Next slide. It turned out that we weren't the only ones seeing these new cases of diseases indicative of underlying immune suppression. There had been a number of them, but nobody had really put things together until 1981 in June, when the Morbidity and Mortality Weekly Report reported a new disease. The next slide. So it had taken 2 years to get to the point of recognizing that from the time the first cases started to appear. Next slide. And sadly, it took another 3 years before we were able to identify HIV as the cause. 4 years from that time to get a diagnostic test, and we're now 39 years into this and still waiting for a vaccine. Next slide. So at that time, I decided to really focus on HIV as a career, and started out then to try and understand how the body fights back against HIV infection and how it usually loses that battle. And we were fortunate along the way to be able to establish the Reagan Institute, whose mission was to harness the immune response to prevent and cure human disease, focusing on infectious pathogens of global importance. Our strategy was cross-disciplinary collaboration, thinking that if we could bring other fields into the HIV field, we could actually make more significant progress. And we were fortunate to be embedded in this incredible biomedical research ecosystem here in Boston. Next slide. So we've made progress since then. Through Dan Baruch, one of the founding members of the Reagan Institute, we have an HIV vaccine trial underway, an efficacy trial in South Africa, and are quite optimistic about that. Next slide. So in January of this year, I was teaching a course that I teach every year called Evolution of an Epidemic. Next slide, where basically we bring 25 Harvard and MIT undergraduates to the heart of an ongoing epidemic to use HIV as a model to understand how an epidemic emerges, how clues from patients guide discovery, and how policy and advocacy influence the course of an epidemic. Next slide. So here we were visiting with traditional healers, and the two people in this slide are quite significant to the story. Next slide. On the left is one of the MIT undergrads who had just returned from Wuhan and was getting text messages daily from her family saying that something terrible was happening there, a new pneumonia, unclear cause, and people were getting hospitalized left and right and dying. And on the right is Diana Brainerd, a former trainee of mine, who's the senior vice president for HIV and emerging viral diseases at Gilead. She was getting, I invited her as a teacher, and she was getting daily calls from China asking her to release a little known drug at the time that they had developed for Ebola called remdesivir. It was clear to me from the discussions that they were having and relating to us that this had all the markings of an emerging pandemic. Next slide. So thinking about our mission, goal, strategy, and location, as I was flying back from South Africa, I just decided this is something we need to get involved in. And our initial focus had been on HIV and TB, but certainly we need to apply what we could to this new problem. Next slide. So what was this disease? Pneumonia of unknown cause. Next slide. And it actually was, in comparison to HIV, incredible how rapidly we went from the first public report, which was somewhere, depending upon your source, between December 20th and 27th, due to somebody recognizing that there was a cluster related to this Huanon market. Next slide. And so it only took a month from the first cases to recognize that it was a disease, as opposed to the 2 years for HIV. Next slide. And this is maybe even more remarkable, that we had identification of the agent and a diagnostic test within less than a month. Just unprecedented speed, and a real tribute to what science has been able to accomplish. Next slide. So the virus is a beta coronavirus. It's an RNA virus with an outer envelope that has a spike protein that is a major target for the host immune response. Next slide. We know that this came from bats originally, probably through pangolins and maybe back to bats in between. Next slide. So it's a zoonosis, and one of many viral diseases that are crossovers from animals to humans. I'd like to just make sure that everybody understands what a virus is. On the left is a bacterium. Bacteria are very different than viruses. They have, in this case, over 4,000 genes for E. coli. And E. coli has enough genes that it actually has all the information it needs to reproduce itself. On the other hand, viruses, shown here as a speck at the end of the arrow, SARS-CoV-2, has 10 genes and can't reproduce independently, but instead have to infect a cell and commandeer the host genes to make new viruses. What's incredible is that the 10 SARS-CoV-2 genes can overtake and direct the 20,000 human genes to play its game. Next slide. So we're now up, I think today, to over 38 million cases worldwide. It's clearly a pandemic that's not going away and is reemerging. Next slide. And if we think about this, fortunately the case fatality rate is low, but transmissibility for SARS-CoV-2 on the third line here is high. On the other hand, the prior pathogenic coronaviruses, SARS and MERS, had much higher case fatality rates, but much lower transmissibility. What I worry about is not just this pandemic, but the next coronavirus, which may have the transmissibility of SARS-CoV-2 and the case fatality rate of MERS, which would truly be an existential threat to humanity. Next slide. So the disease is partly caused by an overexuberant immune response. And you can see that here on these histologic slides. On the left, the normal thin-lined air sacs in the lung. On the right, this tremendous inflammatory influx that results in decreased passage of oxygen into the bloodstream and is the hallmark of COVID-19 pneumonia. Next slide. It's not just the lung that gets infected, but there are a number of other organs that get infected, and that's why cardiomyopathy is part of this. This is why coagulopathy is part of it, and the gut is also part of the problem. The body fights back. Antibody-producing B cells recognize virus and produce these antibodies that can bind to SARS-CoV-2 and specifically prevent it from attaching and entering cells. Next slide. And so if we think about what how immunity is generated following infection, we have somebody who's naive, has never seen the virus, they get infected. Next slide. And what that does is it elicits an antibody response that ideally clears the virus and leaves somebody with long-lasting immunity. Next slide. Unfortunately, the virus itself, in the case of SARS, causes immune system damage. And because of that, there are very weakened immune responses that are generated. And our belief, based on, I think, compelling data now, are that these weakened antibody responses are short-lived, and whether we would get to herd immunity of any lasting durability from natural infection, I think, is a big question. Next slide. On the other hand, let's think about what we're trying to do with a vaccine. Rather than letting the virus do its damage while the immune system's learning how to recognize it, the concept behind a vaccine is basically to take a part of the virus, for example, the spike protein, and expose the immune system to just that, have it learn how to make an immune response, and then have that immune response be ready. Next slide. When the virus when somebody gets exposed. So here you are immunizing somebody with just the spike protein, you generate antibodies, and the next slide shows that basically you end up with immunity. And now in the next slide, you can see what's different about this. Now, because there's not immune system damage, we hypothesize that the durability of immune responses will be much better because they will have been generated in the context of a functioning immune system, because the virus will not, it's not live virus that generated this, it's just the spike protein. Next slide. There's another way to confer immunity to somebody, and that is that an infected patient, once they've recovered, can have, next slide, some antibodies that are broadly neutralizing, and by identifying and cloning the B cells that made those antibodies, next slide, one can produce those antibodies in large numbers, adoptively transfer them to an uninfected patient, and achieve immunity with the best of the best antibodies available. And this is what President Trump received as a compassionate use therapy using the Regeneron product. And Lilly also has a product, and there's some others that are on the way. Next slide. So if we think, though, not about adoptive therapy, but about vaccines, there are a huge number of vaccines already being developed, over 200. 43 or more of these are now in human trials, 11 of them in phase 3 trials. None has been given full approval yet. There are some limited approvals globally for some vaccines that were generated in Russia and in China. Next slide. And the approaches that are being taken are shown here. One approach is genetic vaccines. This is the Moderna and BioNTech approach with messenger RNA, or Inovio with a DNA vaccine. Another is to take and splice some of the genetic material into another virus, like an adenovirus, so that the spike protein is made by the adenovirus, and that teaches the body to recognize the spike protein, whereas the adenovirus is non-pathogenic. So you don't end up with a damaged immune system. The tried and most used method is to use viral protein plus adjuvant. Sanofi and Novavax are developing those products that are going to be entering into phase 3 trials. And then the Chinese have developed an inactivated SARS-CoV-2 where the genetic material is basically disrupted so it can't replicate, and thereby diminish the damage that the virus could cause while letting the immune system learn how to deal with it. Next slide. So the approach that we've taken is with Dan Baruch is to pivot from our HIV platform to, next slide, turn that into a vaccine that, instead of splicing HIV genetic material into the adenovirus, in this case, you splice SARS-CoV-2 genetic material into the adenovirus and then use that as your vector. Next slide. And it's really, I think this is a great example of just how rapidly things have gone. It took, remember, over a decade to develop this vaccine for HIV, and it's still not been fully tested. And yet, between January 10th, when the virus sequence was released, and the middle of September, Dan had gone already from making synthetic genes to phase 3 efficacy trials in humans, with a promise of a massive scale up and emergency use authorization, hopefully in early 2021. Next slide. So these are the various different approaches that are being used. I think I've mentioned them already. Our hope is that as many of these as possible will go forward. Next slide. But there are a number of key questions. Does the vaccine protect from infection or from disease? You'd like it to fully protect from any infection, but if it could attenuate disease to prevent transmission, that would be, and symptoms, that would be a success. Does it protect against severe or only mild infection? If it only protects from mild infection, that's not going to be that helpful. So that's being tested now in these phase 3 trials. How many doses do you need? If you need just one, as per Dan's vaccine, then you need 7 billion doses for the world. If you need 2 doses, that's 14 billion, a huge number. Plus, you have to get all those people back. So huge logistical problems. Will there be enough new infections to show protection? I don't think that's going to be a problem. How soon can the vaccine be widely available? Scale up is happening for all of these, so that's encouraging. Will it work regardless of age? We don't yet know, and studies in children are lagging behind, understandably, until safety is shown in adults. So children in school are not going to be getting vaccines anytime soon. Does the vaccine require it be kept cold the entire time? The mRNA vaccines require that, and that adds huge logistical challenges for global delivery. How durable is the immunity? We will see, but it's there. Even just this week, there's been report of a secondary infection with pretty compelling data that happens. And with other coronaviruses, there are data that suggests that the immunity lasts 6 to 12 months for other human coronaviruses. So there's a concern about herd immunity through natural infection. And can safety be assured? Well, I believe it can be if the regulations by the FDA are followed. In the meantime, what can we do? Next slide. It's very clear. Well, I'll just thank the Reagan Institute for the support that they've given us. Next slide. What we've been able to do is to pull people together here in Massachusetts to form the Massachusetts Consortium for Pathogen Readiness. I'm happy to be leading that with Arlene Sharp. We've got over 500 scientists and clinicians working together on trying to share knowledge and make as much progress as we can. Next slide. There are a number of donors that have participated in helping that whole effort happen, including Evergrande in China. Next slide. I want to mention that the other thing that we can do is we can wear masks. The experiment has already been done in hospitals during the worst part of the pandemic when these nurses in Boston were up to their elbows in COVID patients. They weren't able to distance, and yet they didn't get infected because they wore masks. Next slide. So a few conclusions that I think people can read here. I would just say that the vaccine development has been progressing incredibly rapidly. I don't think we're going to have widely accessible vaccines until the second half of 2021. We need to have as many of these succeed as possible, and there's still much to be done. And I think that's the next slide. That's the last slide. So thank you very much. I appreciate your attention.
Thank you so much, Dr. Walker. Thank you, Brian, for this first part. Can we prepare to send the video, please? It's a pleasure.
Good morning, everybody. I want to discuss with you this morning about what happens in France with COVID-19 and also a relationship between science and the politician, because I've been nominated 6 months ago as the head of the French Scientific Committee on COVID-19.
Okay.
And I want to discuss on that issue with you. So the first cases in France has been described at the end of January, and with the first death at the end of February, and with the first vague tsunami at the beginning of March. And so the scientific committee was on Macron, and we have a meeting with the President Macron on 12th of March, and we have a large discussion of the lockdown, and the general lockdown was decided on 17th of March. After that, we have the end of the lockdown 2 months at the beginning, at the beginning of May, and we have a long period with a long remission of the virus circulation during the summer, and since September, the epidemic is, I really come back on this situation at that time.
Can I see the video, Ethan?
you have some slide about the lockdown in Paris and what happened also in Paris on the Quai de Seine in July and in August, and you are able to see the difference of the situation, and you can also see the difference on the two maps. One on the left with no virus circulation in May and in June, and the other one, and I will come back on this map after, with a great virus circulation at the end of September. So where we are, the key data, we have approximately 3,000 of deaths in France, and the current situation is we have approximately 800 of cases admission in hospital at that time, and about 150 patients each day which were admitted in intensive care unit at that time. So there is a clearly a great viral circulation since the beginning of September, and it was a real difficulty for me and the scientific committee to convince the politicians in France to see the reality and to see that we have a second wave in France. You have the evolution of several indicators of what happens with the pandemic in France, and you can see finally the most important, the number of new cases each day, and at that time, at the end of September, we have about 30,000 of new cases in France, and as you can see, this number was only about 2,000 new cases at the end of June after the lockdown. So there is clearly a difference, and the three phases: one, the lockdown; the second, the post-lockdown; and the third with the new wave in France. So what about the relationship between science and politics? The scientific committee, we are 12 members with a multidisciplinary composition, virologists, clinicians, specialists of intensive care units, but also specialists of human and social science. And so it's really a multidisciplinary approach, and all decisions are taken together. We have three. I want to discuss about 3 points: the independence of the committee, independence vis-à-vis du politique. What does it mean? It means that we are not in a hierarchy with the President of the Republic. We are scientists and coming from university. We can decide about the subjects that we can treat. So we can also decide and we decide to have this all notes and all opinions and have been public for each of these opinions. The second problem or discussions that I want to have with you is the time. The time for science is not the time for media. Media is 1 or 2 hours. The time for politicians is 1 or 2 days, and the time for scientific and physician is generally several weeks or several months. If you want to have the results of a randomized clinical trial, you need to have several months with you. And this notion of time, when you discuss an issue, we can discuss about that, but the time is not the same. The first problem is the doubt. Clearly, the science is built with doubt, and I think it's really a pillar of the science to have doubt. That's not the case with politicians. When they say you are a scientific committee, so we want a decision, and we want to have a clear-cut decision from you. it's on or off, and clearly science is not an off, on off or off, and clearly there is for this problem a real issue when you discuss with a politician. After that, the role is quite different. The scientific council is involved in the decision, but is not concerned by the decision. The decision is taken by the politician. Clearly, the role is different. And in France, the third medical power was discussed by the press, but it was clearly just an image from the press. But in reality, the scientific council does not take decision. It was the politics that take decision, and you have the same issue also in US. I know. When I come back, what we have done during these 6 months, we have 3 phases. Clearly, we discuss about decision and public health issue, and not really medical issue. And we come back on that problems. We try to have a vision, a global vision for strategy, and we have 3 phases in the collaboration with the politician and the President of the Republic. The first phase was for the lockdown, and you have the different opinions that we have. The second phase was for the post and the end of the lockdown, and the first phase is now, since the end of August, and when we say we have a recirculation of the virus in France, and it spends about between 10 and 2 weeks to have a large discussion with the politician, and also with different people in France, to convince that it was the time to take some difficult decision. And clearly for us, September was a difficult month, because the politicians do not want heard in the first time that the virus was de novo and recirculated in France. This scientific committee was involved in thinking about the vaccine strategy, but was not really involved in the therapeutic and research issue, because we have a second committee in France, whose name was CARE, and it was chaired by Françoise Barré-Sinoussi, our Nobel Prize, and it was directly, this CARE committee was directly involved in the relationship with the Ministry of Research. So what's happening now in France? And you have the map of the situation in France, and as you can see, the problem is in the 20 big cities in France. So what kind of measure and tools, voluntary or compulsory, sanitary or political, national or local? We have some responses to this question, and clearly the response is now to the local and to the territoire, and most of the decision has to be taken by the big cities in France, as Lyon, Marseille, Bordeaux, but it's not so simple, because if each big cities take a decision which is different from the other, you see it's a difficult problem at the national level. What about also vulnerable people? As you know, most of the same issues that in US, with most of death occur in older people, older than 70 years, and also in people in precarity. So it's really a major issue also in France. And after that, we have to be and to clearly say also that the use and the possibility to use beds for intensive care unit will be different in October what happens in March, because we don't want to have an occupation of these beds more than 50% for COVID positive patients. So finally, what we need to control the viral circulation, and I think it's not only for France, but it's also for different countries. We have different countries who succeed to control the epidemic. Barrier measure and mask, and clearly I have a mask at that time, but also test, trace and isolate, TTT, and with also using of a smartphone application. And finally, we have also to target super spreading events for surveillance, and that's really a major issue, and with bars, cruising ships, etc. And in France, we have a large discussion at that time on the role of what happens really in the bars for young people, because we think that a part of contamination occur in bars, but finally we have only 4 or 5 scientific papers to prove that. We have also, and that's a new idea for politicians, but also for our citizens, and to clearly say that for COVID-19 we are in a long-term story. A long-term story is not finished. We probably the story for six months or 1 year more before we have drugs or vaccine. So where we are for, no, I don't discuss about vaccine, but I want to just discuss about some major medical advances that occur, that we have seen during the last month. The first one is we know that most of death occur in older patients. The second is decrease of mortality in severe cases by more than about 1/3, and due to new drugs, and also to the fact that physicians take care with the most efficacy. We know also that the virus is stable, and there is no major mutation, and we observe no major mutation in France. Fourth issue is herd immunity, which is low in France, but I think it's exactly the same level of herd immunity in US, about 5%, 12% in Paris area, similar results in different countries in Europe, and you know that we need a herd immunity about 50% to have a real efficacy at the national level, at the populational level for the virus. About drugs, we have no effect of hydroxychloroquine, lopinavir, ritonavir, remdesivir in a large randomized clinical trial in France in collaboration with WHO and the Discovery trial. And we look forward for monoclonal antibodies, and we have, as in US, a lot of clinical trials in progress. So finally, I can say that innovation is not as a rendezvous for drugs. Innovation is on board for testing with TRODS and antigenemia, for example, test. And innovation is partially in board for application as a Stop COVID in France, but you have also application in US. So to finish, some comments on the relationships between the scientists and politics. Clearly, as I said before, expertise was done by the scientific committee, but decision was taken by the politic. And but if I look of what happens, most of our proposal was accepted by the politician. So clearly there is a difference between expertise and decision, but in such a crisis, there is a a major issue for physicians and scientists to propose decisions which are accepted by the politics. We have also a strong willing from our scientific committee to be transparent for and all publication and all opinions and notes were published, and and also addition to the National Assembly and the Senate, etc. We call for a citizen committee carried by the Scientific Council to have the opinion of different citizens in France, but such a committee was not accepted by the French government. And finally, we have during these 6 months a constant doctrine of personal citizen responsibility and choice, nothing imposed, and voluntary choice for each measure. And I think that's clearly was an important issue that was also accepted by our politicians. Next slide. And finally, at that time, I want to discuss with you the last slide. That's just later from Richard Horton in the Lancet last week, and which calls Science and the Breakdown of Trust. And clearly, at that time, we have a major issue for COVID, and the COVID-19 systemic is entering in the most dangerous phase, because there is tension between scientific decision, politic decision, and the acceptability of this decision by the citizen. And this is a major issue. For example, in France, we have a large discussion from different big cities about the fact that we can close the bar, for example, and the restaurant, but it's not only for that. And clearly at that time, the acceptability of the measure is a major issue, and we have to come back not only to science, but also to human and social science to discuss with our citizens. So to finish, Richard Horton in the editorial says that Sylvain Todorov, who is a philosopher which think about the relationship between politician and citizen, says that debate rather than consensus characterize our modern era. We should not be afraid of disagreement, debate rather than consensus.
Thank you, Professor Delfraissy, for a very comprehensive Thank you, and I will retrospectively introduce Professor Delfraissy. We are operating live. It is a little bit challenging. Professor Delfraissy is the chairman of the National French COVID-19 Scientific Advisory Board, advising the President of France and the government. He's also the chair of the National Ethics Committee. Gives him, I guess, a very interesting perspective. He had a last-minute conflict, as you saw. The recording was done on Friday, the 10th of October at 10 a.m. Eastern, 4 p.m. in France. I would like to thank Professor Walker and Professor Delfraissy for coming together live with myself and Brian Malkin to organize, coordinate for this meeting. We will now move, my excuses to the audience because of we are going over.
We will now move to our next panel, panel number 2. This panel is moderated by my good colleague, Dr. Cécile Le Camus, who is Executive Medical Director and Medical Lead at Vertex Pharmaceuticals here in Boston. Cecile's participation here is especially relevant because she's a former emergency room physician and she is a pharmaceutical physician who cares very much for patients. Cecile, it's all yours.
Okay, thank you very much, Shahin. So as we've heard from our previous distinguished speakers, COVID-19 is definitely a formidable challenge posed to the public research institutions and the biotech pharma industries. The Boston Business Journal has reported that some 30 companies in Massachusetts alone are focused on COVID. And for having accessed clinicaltrials.gov this month, I have seen over 1,000 interventional studies in this field. With that, I'm thrilled to introduce Priscilla Douglas and Sophie Karnowski, who will be discussing how this pandemic is challenging our communities faced with leadership deficiencies, confusion, and ambiguity. Priscilla, after experience as government and pharmaceutical executive, today serves on the board of the Boston Museum of Science, and St. Jude Children's Research Hospital. Priscilla is also an author and an executive coach. Sophie is a senior partner with the Gurnett Point Capital, a healthcare fund, investing in long-term capital and supporting entrepreneurs to build the next generation of companies, prior to which Sophie has held a number of executive roles within pharma. Starting on the leadership front, Priscilla, with your over 30 years of experience observing and coaching leaders, can you explain or discuss what you've noticed about their behavior in the pandemic?
Thank you so much for asking. In this, the world has just been turned upside down. And one of the things that I think you all know is that nature abhors a vacuum. And into that vacuum, a number of different leaders are emerging with very distinct leadership styles. And the big difference that I've noticed in this COVID age is that these leaders are not only mindful and collaborative, they're inviting new people into the conversation, which is a significant difference from in the past. And I think if you just look at the scope of the collaborations and the reach that they have, that you can see that those voices really, really matter. Another big thing that I've noticed is that people's idea of change has clearly been disrupted. And you think about the word disruption, where rupture, right? Disruption, things have been broken, changed significantly, and there is no going back. So just in May here in the United States, when we think that things are gonna calm down, on May 25th, we witnessed the death of George Floyd. We saw the life pressed out of him. And in a situation like that, that worldwide, that affected all of us. And it's something that you can't unsee. So when we think about change, what I want you to begin to think about is not the traditional type of change, but think about the butterfly effect. The term, is used in weather when we think about what happens in Brazil, what happens around the world, but also it came out of the term Ray Bradbury's novel in 1952. Let's take a look at the first slide, because, the world stopped. And in this pandemic, do we have the first slide? Thank you. The world stopped. When we think about everything about our lives was put on hold, not postponed, canceled. Ambiguity, all of our plans were up in the air, all the things were upended. And in this cancellation, the cancellation has a spiral effect, of course. It's a constellation of events in terms of the flights were canceled. People were in their homes, which shut down the airports, which shut down our supply chains, which disrupted everything. It's a far-reaching global effect. Our economic systems upended, our social systems upended by the cultural change. There is no return to normal. There is no return to normal. But as you can see in the next slide, our leadership model has been upended. Typically, managers have been following, really worldwide, the McKinsey 7S model. You think about what is our strategy? What's the structure that we need to put in place? And what are the systems that hold that structure together? And those were called the hard S's. And almost all good organizations had that. And then they said, well, then you need to focus on the softer issues, the values, the skills, and the staff. But here's the thing. In this pandemic, next slide, what we value in a leader has changed significantly. And in that change, we're now thinking about the norms that people have. We see that the essential workers, frontline workers, the humanitarian effect. The next slide. We're experiencing what I'm going to call is collaboration 2.0, because we have to go as far reaching as we can in our ecosystems to bring people in. And AstraZeneca, our CEO, I just love the way that he approaches it, because casual intensity is kind of the style. You really can't take yourself too seriously. You have to be open to questions, and you need to take bold moves. Next slide. These disruptions lead to a transformation. And here at home in the United States, we watched Andrew Cuomo, very different style from Pascal Soriot, very different style, but he too was putting together coalitions and consortiums and bringing new people together, different people together. You saw regional collaborations, New Jersey, Connecticut, New England, the whole New England coast had to band together in order to get PPE and supplies, to put in place a new supply chain. And he also got us to think about what are the new norms. And these norms is what this new generation of leaders are really thinking about as a way to envision the future. Tough and smart. Next slide. So as you begin to think about, I'm going to just give you some ideas so we can get to the dialogue. One of the things that these leaders are doing is, they're reacting, they're relating to new people, they're recalibrating how they're getting things done, they're reimagining. But last slide. We all know that this isn't a one-of-a-kind event. A Harvard professor has been studying these, and when we look at it, there's no such thing really as a black swan. Look at the amount of disruption and change. And the new generation of leaders, we can go back, that's the last slide. This new generation of leaders is not ignoring these trends. So we're humanity, taking a look at inviting new people into the conversations, collaborating in an open way with empathy and with humanity. Just some ideas for you to think about as we look at the leaders who are emerging in this COVID environment.
Thank you, Priscilla. This was pretty inspiring. And so kind of shifting from leadership to finance, Sophie, and with what we've just heard from Priscilla about collaboration and disruptive leadership, how does this really translate into your investor world?
Thank you, Priscilla and Cecile. This is a very interesting topic, to say the least. And as often, you prepare slides and then the speaker before you inspires you to say other things. So I just think it's quite a fascinating moment. I was delighted to hear Professor Delfresi. I was head of Roche in France when we had the H1N1 pandemic. And what was very interesting is back then, it was for sure that another one would come one day. And there was a whole discussion about how would we be ready. So in a way, it's nothing to do with a black swan event. It was highly expected. And the question was just when and how bad would it be? And I think this is, but nevertheless, none of us was prepared. And I would be, say, really humbling myself when I heard what was going on in China and Hong Kong early in the year. Not in a moment did it cross my mind that this virus was going to take an airplane and come to visit us soon. So, it's always easy after the fact to think that we should have done things differently. I have to say people have said really silly things around the world, and I was one of them. So this is a humbling moment, I think, for everyone and for me, for sure. But let me turn now to the financial situation of our industry, and then I will talk about what goes on in the companies. And that first slide can already show you that, it's been a really good year for pharma. It's quite stunning, but best year in financing in 2 decades. Blueing, the 18 banner. And of course, you have a large amount of financing that's coming from COVID companies, for sure, between Moderna raising 1.3 billion, BioNTech, and all of that. You have 350 COVID trials ongoing and companies behind those, not to talk about what's going on in the vaccine world. But at the end of the day, 922 biopharma financing, and it's quite remarkable. And even if a quarter of them are around COVID, the rest is not. So it's been a very healthy year for, after a little blip for a moment, it's been a very healthy year for pharma. If you're going to go to the next slide, please. So what about the indexes? You can see we had our moment of dipping the whole market together with the S&P and everyone. But after that, the biotech and NASDAQ went back very strongly. Now we see a lot of volatility, but it is a very strong year in terms of valuation. And that makes, of course, any deal more challenging, and we'll see who has been winning in such a healthy business environment. Next slide. So what about the IPOs? We are talking about a record here, which is quite stunning if you think about it. So IPOs in the health tech field with One Medical and Oak Street and all of that, but you also have had IPOs of a Chinese company, of American companies, and in Europe, it's been really lagging. We haven't seen, I think there is only one IPO. So this worldwide energy that was injected into the biotech world has unfortunately not benefited Europe. 19 IPOs, only one in Europe. It continues to be disappointing because the science is great overseas as well, but unfortunately this has not showed yet. Next slide. So M&A, of course, with a very, very strong valuation, not a surprise that the M&As have been more sluggish. We've had the remarkable Gilead acquisition of Immunomedics, 21 billion. This is quite a great deal. We've had smaller ones with Momentum, Myocardia between BMS and J&J, and a very intriguing one with Nestlé acquiring Aimmune, peanut allergy treatment. But overall, I think that, with such a strong level of valuation, not a surprise that the M&A have been more on the sluggish front. Next slide. And as I said earlier, very strong venture cash raised, and it's quite interesting. So we are going to be, at the record level. And what is also very clear is that companies have also been extremely careful on how they do their spending, and that leads me to my next point, which is now let's talk a little bit about what goes on inside the companies. And I don't have a slide for that, but I just think it's really important to also talk about this. So people today are worried to be moving. It's been challenging to hire people and challenging for people to take bold career moves. Because the valuations are strong, people are more tied to their companies. It's quite remarkable, however, that companies have been able to navigate the challenges of complex clinical trials, recruitment, continuation, even launch in an elegant way, preserving the cash, having a good dialogue with the FDA. I think we have a lot of companies in our portfolio who've had FDA interactions, and we've been really impressed how the FDA is not only handling all this COVID pressure, but is also having the right type of a dialogue with pharma companies to help them push forward. So that I think is extremely important. Some analyses are coming out. I think it was in the past couple of days, a review of the death rates worldwide that in many countries has been increased by 30 to 40%. And it's not all COVID related, it's also related to people not having the right type of care and preventative treatment. So this pandemic is not a virology thunderstorm, it's also a healthcare thunderstorm in general for elderly people for sure who are at risk, but also the cardiovascular people, the emergency care, and the preventative treatment. So a lot is going on right now. What happens in the companies is you have to be very careful on the workload and the pressure of people. At the beginning, everybody was like, oh, there is no way I'm showing my face on Zoom. Now nobody really cares to even show himself in pajama on Zoom as long as the work is getting done. So there is, this casual intensity you talk about that Pascal has. And by the way, Pascal always had it because I worked for him way before there was it. It is just his style. And I have to say it's just really special. But everybody now is getting to be that type. Let's get things done, roll up our sleeves, work off hours, get things where they need to be so that we play our role in our industry. I think when so much valuation upside is happening, it's really key for us to remember our mission. For example, I think we can safely say that all those leaders who are involved in COVID, maybe some are thinking about making a lot of money, but most of them are thinking about making a huge difference in the world. And I think this is what should get us out of bed every day. Priscilla touched on it, but this is really the time where have and have-nots are becoming incredibly visible. It should put us all at unease. And remember that we have to fight for drug access. I think that when someone in a leadership role at the government level gets saved because industry makes available drugs ahead of time, that shows how critical we all are and how much we have to fight, not only for our own little territory, our own little products, but also for industry in general and to make sure we can continue our mission. So it's a time for connection. It's a time for making ourselves available. And I have chosen to take it as energizing and motivating. And whenever I get scared, I look at what others are going through and I remember to be, courageous and, out there to help. So this is just this event participates to that. So thank you for making it possible, Shaheen, Soreya, and everyone.
Thank you very much, Priscilla and Sophie. I'm unsure if we have time for questions, so I might kind of squeeze one in. Putting on the patient hat, and I think it's a question for both of you really, is, and listening to what you've said about needing to change the leadership style, there's maybe one other thing, back to the patient, is how as leaders can we show the silver lining in the pandemic, which you were alluding to, Sophie? It's time for change, it's energizing. How do we go about showing this?
One of the things that these leaders do is that they create a future for people to live into. So you know that in a lot of the work in terms of being mindful and for people to really be able to envision that they will get through this and there's life beyond that, that not only happens for your well-being, it happens for the well-being of the organization. So it's a different way in terms of thinking about a narrative and a strategy, which is to create a future. And what is that? Who's in it? And to make that future as inclusive and relevant to the people that are affected.
I have, spent a lot of time talking about the silver lining to my son. I have a 20-year-old child, and he was like, "What the hell is going on here, Mom?" And I have to say, I have tried to do so many deals in antibiotics and virology, and getting little traction, finding it very challenging. As I said, the H1N1, to get people to be aware and worried and realize that you really need to protect people was a real challenge. So I think there is an avenue for a better world, better protected healthcare worker, better system for taking care of people who are challenged because of where they are in the society, another way of interacting with elderly, and a better way to protect ourselves. The planet is not open for business, and we can fly and eat anything and do anything. There is some limitation that we have to put to ourselves. And right now we're forced to it. But better treatment of our planet, because at the end of the day, the way we treat the planet makes those viruses possible. So I am an optimist, but it will take work, and it will take people standing up and trying to do my own share of it.
Thank you so much, Sophie and Priscilla, and sharing your positive attitude. I'll just conclude. Although terrible and challenging, this pandemic may also be our chance to disrupt the way we do things and learn to work differently in healthcare through collaboration, trust. We've heard about trust earlier, long-term partnerships, and disruptive leadership. I'll keep that with myself very close too. With that, I'll hand it over to Shane. Thank you.
Thank you for an outstanding panel, both inspirational, a lot of data, and a lot of food for thought. We tried to prevent COVID in the first panel. We looked at leadership and financing in the second panel. What if you have COVID? What happens then? You need a treatment. This is why this third panel is essential. It is going to be co-moderated by Dr. Vanessa El-Harar, who is Vice President for Vaccine Strategy at PPD Biotech and formerly from the NIH, National Institutes of Health, and Dr. Nicola Lamonica, who is Senior Director, Infectious Diseases Scientific Innovation in the Johnson & Johnson Group here in Boston, and himself a highly qualified virologist. The floor is yours.
Thank you, Shaheen, for the introduction. On behalf of PPD, my co-moderator, Nicola Lamonica, and myself, we're very pleased that all of you have joined us today to discuss the current status of therapeutic development for COVID-19, and also translation of that research to actual frontline medical care received by patients. So our first presenter is an accomplished infectious disease physician and researcher who really needs no introduction. I'll introduce him anyway. Dr. Dan Kuritzkis, the Chief of the Division of Infectious Diseases at Brigham and Women's Hospital and also Professor of Medicine at Harvard Medical School, is here today to speak with us about therapeutics for COVID-19. Dr. Kuritzkis, I'll give you the floor.
Oh, thanks very much, Vanessa. It's really been a pleasure listening in on this incredibly interesting symposium all morning or afternoon. In the next 10 or 12 minutes, I'm going to give a very high-level, rapid-fire overview of where we stand in the development of therapeutics for COVID-19. If I can have the next slide, please. These are my disclosures. Next slide. So I will cover four topics: remdesivir, corticosteroids, convalescent plasma, and monoclonal antibodies. Next slide. Remdesivir is an RNA-dependent RNA polymerase inhibitor that has in vitro activity against a range of viruses, including Ebola, SARS, MERS, and now, of course, SARS-CoV-2. Against SARS-CoV-2, it's shown submicromolar EC50s in vitro and has also been demonstrated to have activity both for prevention and treatment of SARS-CoV-2 in the non-human primate model. Next slide, please. The most compelling data in terms of its clinical efficacy come from the ACT-1 study, a collaborative study done through the National Institutes of Health in the United States. This was a placebo-controlled, double-blind, randomized clinical trial in hospitalized adults with COVID-19 pneumonia. Participants were randomized one-to-one to receive remdesivir or placebo, with a primary endpoint of time to recovery within 28 days. This is an important point because there's a difference emerging in perspective about whether recovery time or prevention of mortality is the most appropriate endpoint in such clinical trials. The preliminary analysis was conducted after slightly more than 600 participants had attained recovery. What you can see in panel A is that looking at the study population overall, there was a statistically significantly faster rate of recovery among the remdesivir recipients compared to placebo. This difference, however, was not significant when examining just those participants who did not require oxygen, nor in the sickest participants who were receiving either high flow oxygen or mechanical ventilation. But it was clear, even more pronounced if you limited the analysis to those who were receiving supplemental oxygen. Next slide, please. Then there were two trials conducted by Gilead, the SIMPLE trials, one in severe disease that compared a 5-day and 10-day course of remdesivir, and one in moderate disease that looked at, again, 5 or 10 days, but also included a placebo arm. The severe trial did not have a placebo arm. However, there was a contemporaneous group of patients at centers not participating in the trial who were recruited by the company in order to provide comparative data, so a form of control, but not a randomized control. And when the outcomes in the pooled remdesivir group from the SIMPLE severe trial were compared to these untreated or standard of care participants, you can see that there was, in fact, both a significantly greater recovery at day 14, nearly 75% versus just under 60% in the control group. And there was a significantly lower mortality rate, about 7.5% versus 12.5%, suggesting a survival benefit for remdesivir, but not really proving it because this isn't a placebo-controlled trial. Next slide, please. And then these are the outcomes in the moderate disease trial, which are somewhat mixed. You can see that at day 11, the shorter course arm, 5 days, had a significantly greater improvement, but not the longer course, which doesn't make an enormous amount of sense. Then at day 14, both arms have similar recovery rates, but at day 28, things are slightly flipped. The longer course shows significance and the shorter course does not, or just misses being significant. Overall, these results are somewhat ambivalent or equivocal, and therefore neither the DHHS guidelines panel nor the Infectious Disease Society of America have endorsed the use of remdesivir in people with moderate or mild disease, but strongly recommend remdesivir as standard of care in people with severe disease. Next slide, please. Let's move on now to corticosteroids, and the main study in this regard was the recovery trial. This is actually a multi-arm study being done through the University of Oxford in the United Kingdom, where participants are randomized to one of several different arms or a shared control arm, focusing just on the dexamethasone trial. This study randomized participants either to dexamethasone or usual care. There were about 2,100 in the dex arm and 4,300 in the usual care arm. And what you can see is that overall, there was a significant reduction in the relative risk of mortality now among all of dexamethasone recipients. However, if you stratify that analysis based on severity of disease as reflected by the requirement for oxygen or mechanical ventilation, some very interesting differences emerge. First of all, it's clear that the largest benefit is observed in those participants who required mechanical ventilation, with a 35% reduction in risk. A more modest but still significant reduction in risk among those requiring supplemental oxygen. However, in participants who did not yet require oxygen, there was actually a trend towards an increased risk of death by use of steroids. Now, how might this be explained? It could be that early in disease, premature administration of corticosteroids suppresses a necessary immune response, whereas with more established disease, when you're beyond really the viral phase of illness and more into the inflammatory phase, you are now damping down an immune response that has now become more harmful than helpful. Next slide. And I should have mentioned that those results really led the WHO to endorse the use of steroids in severe disease and to avoid their use in early disease. Well, what about convalescent plasma? Convalescent plasma from survivors of COVID-19 infection may contain high titers of neutralizing antibody, and it has been shown to have some benefit in treating avian influenza, H1 influenza, and MERS. And pilot studies and uncontrolled trials suggest a possible benefit. Next slide. These data from the expanded access protocol that was administered by the Mayo Clinic in Rochester, Minnesota, show the data from more than 35,000 patients who received convalescent plasma. You can see that more than half of them were ICU patients and more than a quarter were intubated. There are several important points before I go into the actual results from this analysis. First, there is no control group. The comparisons are either early versus late administration and administration of low, medium, or high titer plasma. Second, in most cases, plasma titers of neutralizing antibody were not predetermined before the participants received plasma, and therefore there was the ability retrospectively to analyze the impact of neutralization titers. The 7-day mortality, when you compared early recipients, that is transfusion within 3 days of hospitalization versus 4 or more days later, was substantially reduced with about 8.5% mortality versus nearly 12%. So a roughly 25 to 30% reduction in mortality. And similarly, the 30-day mortality was just under 22% in the early treated group versus 27% in the later treated group. Again, about a 25% reduction in mortality. Now, if you look at the difference in titer, you can see that for the 7-day mortality, there is a trend suggesting that mortality was lowest in the group receiving high titer and plasma. That was just marginally significant by the confidence limits there, looking at the relative risks. And if you break that down further, as in the upper right of the graph, and you look at those who received high titer plasma within 3 days of hospitalization, there was clearly a significantly greater reduction in risk of mortality compared to the other groups. And the lower set of graphs shows the 30-day mortality, and those differences don't quite reach statistical significance. So next slide, please. So to summarize this convalescent plasma, anecdotal evidence and uncontrolled trials suggest that the convalescent plasma may provide some benefit in severe COVID-19. The benefit appears related to early administration of high titer CCP. there's little or no benefit in critically ill or intubated patients. There are still randomized trials ongoing, and it will be critical for those studies to be completed so that we can have really robust data. And in the meantime, the IDSA guideline panel recommends that plasma should only be used in the context of a clinical trial. And similarly, the DHHS COVID-19 guidelines panel says that there are currently insufficient data to recommend either for or against the use of convalescent plasma, and that this therapy is not yet considered standard of care. The next slide. So let me finish with a couple of words about monoclonal neutralizing antibodies, which have been very much in the news recently. Human monoclonal antibodies are able to neutralize SARS-CoV-2. They target the S protein, often but not always the receptor binding domain of the spike. They can be genetically engineered to combine multiple specificities or to extend the half-life and have potential utility both for treatment and prevention of COVID-19. Next slide. Speaking first about the two Lilly antibodies, CoV555 and 016, the BLAZE-1 study is a multi-step trial from which preliminary results have been released by press release or online posting. This study enrolled symptomatic outpatients with COVID-19 and examined the impact of both CoV55 by itself or in combination with the 016 antibody. But there is no study so far of just 016 alone, which is important when interpreting the combination data. The study showed that for the monotherapy arm, the change in virus load endpoint at day 11 was met in the 2,800 milligram group, but not in the higher or lower dose groups, a 700 milligram or 7 gram dose. ER visits and hospitalizations were lower in the recipients of the antibody than in the placebo group, and even more so in the combination arm. There were no study-related serious adverse events. There were viruses with putative resistance mutations that emerged in 8 treated and 6 placebo participants, and additional trials are ongoing. And now we know that the company has also submitted an application for an emergency use authorization. Next slide. I'm sorry, these data show the actual changes in virus load and the differences in outcome. And you can see that a blue line there is the combination arm. I won't spend more time here since I've already summarized these results. Next slide, please. Now, the Regeneron CoV-2 cocktail has been studied in a seamless phase 1-2-3 trial from which preliminary results are available for the early phases. In this study, 275 outpatients with COVID-19 were randomized to receive either 8 grams or 2.4 grams of the cocktail or placebo. Serologic status was highly correlated with baseline virus load. That is, those who were still seronegative at enrollment had the highest titers. And there was a half-log greater reduction in the level of virus recovered from nasopharyngeal swabs in participants who received the high dose as compared to the lower dose, which had only a 0.23 greater log reduction versus placebo. These differences were all slightly greater if you confine the analysis to those participants who were seronegative at enrollment. Among the seronegative participants, the median time to symptom alleviation was 13 days in the placebo group and shorter in both the high dose and low dose groups, but not significantly so. It's interesting when you think about the convalescent plasma data where earlier administration is more beneficial. It may be that for antibody therapies generally, treatment before the host develops their own antibody is effective and maybe less so once the patient's own immune response is already kicking in. Both the high and low dose of the Regeneron cocktail were well tolerated, and again, additional inpatient, outpatient, and prophylaxis trials are ongoing. Next slide, please. So to summarize the story for the monoclonal antibodies, numerous monoclonals have been produced. They appear to reduce virus load, protect against infection, or reduce lung injury in animal models. There are multiple human clinical trials that are ongoing. The preliminary results for both the Lilly and Regeneron antibodies are encouraging. No safety concerns to date, but we really need phase 3 data to demonstrate clinical efficacy and applications for EUAs from Lilly, as I mentioned, and also from Regeneron are now under review by the FDA. Thanks for your attention, and I'll turn this back to our moderators.
Thank you very much. I think it's my turn to, first of all, welcome you all to this conference and introduce the next speaker, who is Professor Karine Lacombe. Professor Lacombe holds a professorship at the Sorbonne Université, a medical school, and is head of infectious diseases at the Saint-Antoine Hospital in Paris. She has an extensive clinical experience in the treatment of HIV and HPV patients, and also obviously she's been very much involved in dealing with the pandemics. And her presentation would be related to giving you an update on the COVID-19 situation in France. Unfortunately, she was not able to attend the symposium in person, but has provided a recording of her presentation that I think now Shaheen will share.
Hello, everybody. I'm really happy to be here today. I'm Professor Karine Lacombe, infectious disease specialist, and I'm heading the Department of Infectious Disease in Paris. And since the beginning of the epidemic in February, I've been very deeply involved in the management of the crisis. And I wanted to share with you what we have done in France in terms of medical care being in the front line. So first of all, I really wanted to tell you that at the beginning, the COVID-19 could have been a great story of tropical forests in China. And you have here pictures of those great forests where you can find those little animals, the bats, and here the pangolin. But in fact, it's not a story of trips in China. It's more a story of globalization, deforestation, and crossing the species barrier. So first of all, to set up the scene regarding the COVID-19 cases globally, more than 36 million cases of COVID-19 have been gathered till up to the 10th of October. And when we look at the dynamics of this epidemic, you can see from this picture that there is a kind of stagnation in the Americas and a relapse in Europe. And Europe is a deep blue at the bottom, a slow but consistent progression of disease in Asia and the sporadic cases in Africa. And you have here the number of notification per country, and you see that the countries the most affected by the epidemic are Europe, and especially Western Europe, but also the Americas, as you all know. Regarding now the dynamics of death from COVID-19, you see that in the Americas, the death toll is really high, more than 1 million deaths so far globally. And if we look at Europe, that you can see with deep blue, we had a high peak of death in March and April, and then a very small number, quite different from what is going on in Asia and America. But you see that at the beginning of October, we see a rise in the number of deaths, and we are very, very much afraid about the second wave that is currently hitting Europe, and especially France. And some instructions from our government, and maybe we're going to have to go back to the shutdown. So regarding now Europe, an update on the COVID-19 situation. You see here, maybe better than in the previous slides, the situation in Europe. About almost 4 million cases have been reported since February 2020, with almost 200,000 deaths so far. And you see the situation reported by the European CDC at the bottom left of this slide. You see that France is really much hit by the epidemic and by the relapse in the epidemic. And you can see that especially with the figure on the top right, where you see that all Europe and especially France are reported a lot of cases now. But you have to bear in mind that in February, March, and April, we were not screening so many patients as we are doing right now. So that's why the number of cases now is very high, but it's also very high because the epidemic is really thriving at the moment. I think it was really important to share with you the data we have on mortality in Europe in 2020 compared to the baseline and what we think should have been the normal mortality rate when compared, for example, with last year. And you see that in 2020, we had a clear excess of mortality in February and March. And we are kind of right now in September, October. So at the end of the graph, you see that that's exactly the same rate of mortality than last year, sorry. So we'll see in the coming month if we see an excess in the death in the coming weeks. So regarding France being in the front line, I wanted to share with you a few challenges that I have identified that we had to face, especially during the heart of the crisis in March and April, and that may still be challenging right now in October. The first challenge we had to face was the problem with the identification of patients who may rapidly deteriorate upon arrival from the emergency room. We all know now the risk factors for severe cases of COVID-19, which are based on the age and underlying comorbidities such as cardiovascular disease, diabetes, chronic lung or renal disease, cancer, and some other presumed risk factors that are listed here. But however, even when we knew about those risk factors, we still had patients arriving on room air in the morning in the emergency room and who got intubated in late afternoon. So we did not know how to avoid direct transfer from ICU from emergency room to ICU, where the number of beds is limited. And in March and April, we had 5,000 beds for the whole country. We had been able to go up to 7,000 beds, but it has been really, really difficult in two regions of France, the east of France, close to Germany, and the Paris area. And you see here a picture of my nurses during the epidemic in April. So besides the traditional risk factors, because the science is bringing answers to the questions we had in April, we have identified immunological predictive factors of disease severity. And I wanted to highlight those three papers which are reporting the immune signatures associated with a poor prognosis, or serological signatures and data regarding association of inflammation due to COVID-19 and the activation, for example, of the C5a, C5a1 axis, which is now a target of specific therapeutics. And I also wanted to highlight this very important paper, which is an international collaboration paper, which has shown that there are inborn errors of type 1 interferon immunity in patients with life-threatening COVID-19. So maybe by targeting interferon alpha and increasing the possibility of this, we will be able to face the challenge of managing severe COVID-19 in patients with comorbidities. The second challenge was how could we do without non-efficient therapy for many weeks? You know that there has been a lot of controversies around hydroxychloroquine and remdesivir, and especially in Europe. And a lot of drugs have have had a compassionate use like tocilizumab and convalescent plasma, and we had consolidated data regarding the impact of steroids only after the peak of the epidemic in France. And we very soon saw that there was an impact of supportive care on the mortality, like anticoagulation and oxygen delivery. And we had those data only from observational settings. That's why we, at the time of the peak in March and April, we had some daily and then bi-weekly multidisciplinary meetings with internal medicine, infectious disease, ICU specialists, the pharmacists, and the clinical team as well. And what you see here, the little cartoon with malade, which means sick in French, and very simple because we had this huge debate in France, which has led to a lot of money invested in proving that chloroquine was not efficient. You see that this is a very simple cartoon to bring some, some laugh from the people who thought that when you were sick, you just had to take chloroquine and then you got cured. And of course, it was not that simple. And following this second challenge, we have established local guidelines. Well, APHP, the biggest hospital structure in France, more than 50,000 people working at APHP. And we have set up all the data we know about the treatment, and then we have established that there is a priority to include patients in clinical trials, but in all cases, all patients in need of oxygen should be treated with dexamethasone for 5 to 10 days. And the use of tocilizumab or remdesivir should be done according to the inflammatory status. Same for convalescent plasma, that we restrict to the patients with B-cell depletion. And there is a huge emphasis on supportive care like anticoagulation according to the dimers level and oxygen delivery, including high flow and the prone position, even outside of ICU, because using high flow or the prone position really prevent people going to ICU where the number of beds is limited, as I told you a little bit earlier. Another unexpected challenge that we had to face during the peak and that we see again now during the second wave is a neuropsychological injury associated with COVID-19. And I would like to share with you the two stories we saw with two different patients. The first one where we see that there was a decreased awareness of severity of respiratory deterioration. You see, for example, this is with this 52-year-old male who was washing himself at the basin without oxygen. Also, we had called the intensive care specialist for an advice after rapid respiratory deterioration. And you see here that this patient had a polypnea of 35 per minute with a need of oxygen of 12 liters per minute, which is really high, only to maintain an oxygen saturation above 92%. So there is a complete disconnection between the status of the patient, the clinical status of the patient as assessed by a doctor, and the clinical status that the patient had of himself, so the perception of its own status. And then with the second case, you see that a lot of patients had problems to identify dyspnea, hypoxemia signals, and a kind of emotional blunting. And that was a 61-year-old male who was on the verge of being intubated. So that's very, it meant that he had a huge hypoxemia. And at that moment, he was not even anxious of his own status. He was just asking the nurse to call his daughter, so sorry, not her daughter, but his daughter, so the daughter would not forget to call the mechanic for his car. So you see that a complete disconnection again between the state of the patient and the perception he had of his state. And finally, after discharge, we saw a lot of patients with a mood instability with episodes of great emotional distress and irritability that we called a post-traumatic stress disorder. Another unexpected challenge was maintaining other infectious disease activity in the context of an all COVID-oriented hospital activity. So you see here, this is a picture of the entrance of my infectious disease unit, where we had 42 beds, usually all kind of infectious disease. And for the first time of our history, all 42 beds were designed, were oriented for COVID-19 infected patients. And that happened to 90% of the beds in my hospital in the center of Paris. 90% of our beds were only for COVID-19 patients. It has never been seen in history. So the situation in infectious disease department, we had to stop all ambulatory care. For example, the STI clinics had to stop their activities as of mid-March 2020. We maintained the daycare hospital activities only for emergency, and we had the disappearance of all other infectious diseases. And the issues here, and you see I've listed only 2 issues, but because that's the topics I'm working on for the last few years. But of course, you can think about all the other issues exactly the same. For example, in cancer, breast cancer, for example, a lot of people have been screened quite late, and local breast cancer became disseminated, within those 4 past months, because the woman did not go for screening. Anyway, regarding my own experience, we had a delayed diagnosis of hepatocellular carcinoma in hepatitis infected individuals, and other cause of undiagnosed acute hepatitis C, undiagnosed acute HIV, etc., and especially in at-risk individuals. And I wanted to highlight this paper very recently published on the indirect effects of COVID-19 on global health patterns. And because I'm working a lot in liver disease, and especially in viral chronic infections, my colleague from Henri Mondor Hospital, very close to my hospital, just published a paper called COVID-19 and the liver-related deaths to come, that we right now have a very high number of deaths due to COVID-19 patients, but in the coming month or even the coming years, we have to be ready to face a high number of deaths of all the other diseases that have not been diagnosed because of the epidemic. So you see that, however, the greatest effect of COVID-19 on liver disease will be indirect and delayed, resulting in the impending global economic crisis. So I would like to thank you for attending this presentation, and I really try to share with you what have been our daily challenges during the crisis, and what is still to be faced in the coming weeks. And this is just to show you that my team and I have written a cartoon book to share what we have been living for the past weeks. Thank you.
Okay, I think we have time for a couple of questions, and I would like to perhaps ask Professor Kuritzkis about his thoughts about how you see the development of neural therapeutic approaches for the treatment of COVID-19 infection and pandemics, in a way, considering the contribution of both a viral infection as well as an inflammatory disease, and how you would think of treating them.
Yeah, thanks, Nicola. That's a really important question. I didn't have time in my talk to mention that there have been a large number of studies of various anti-inflammatory agents, including several studies aimed specifically at the IL-6 ligand access or receptor access, which have been disappointing. We do have at least one marginally successful trial, the ACT-2 trial, combined remdesivir with baricitinib and showed a benefit, but the benefit was people got better 1 day sooner than with remdesivir alone. So I think the underlying hypothesis makes great sense that one should use an antiviral and potentially an anti-inflammatory agent, but we still need to understand which combinations in what sequence and with what timing in order to optimize therapy.
Thank you, Dr. Koritskis. It looks like we have 1 minute left, so I'll take advantage and ask one last question. I'm curious what your thoughts are on the role of an emergency use authorization in the pandemic setting. What do you see as the advantages and disadvantages?
I think an EUA can be an appropriate device when there are adequate data. And I would argue that with remdesivir, once we had data from Act 1, it was entirely appropriate to accelerate access to the drug through an EUA. I'm much more concerned about issuing EUAs solely on the basis of phase 1 and 2 data, which could really hamper our ability to obtain solid phase 3 data that might lead eventually to appropriate licensing or registration of the drug and much broader access to a bigger population.
Thank you so much. And thank you, Dr. Koritskis and Dr. Lacombe, for your time and effort. Wonderful presentations. I will turn it over to Shaheen.
Thank you so much for a fantastic third panel summarizing the treatments. I will now conclude this panel by starting to thank our audience who from Canada to the north to Australia in the south connected today in several different time zones to get more information and more data. Next slide, please. I think one of the good actions, I would say, in these times is to go back and learn from history. And I would certainly recommend everyone this book by Professor Alston is a classic. It has been re-edited and you can learn a lot and it's good food for thought as we are going through this challenge. Next slide, please. Summarizing panels 1, 2, and 3. In the first panel, we had a complete overview of the vaccines in development. This slide has been made from the recent World Vaccine Congress in September of this year, virtual Congress. In the talk with Professor Delfresi, I had 2 questions which we could not show for the sake of time. He is encouraging innovation and the need for new treatments. And he also believes that passage from a directed public health control to a collective public health control is going to be challenging. Next slide, please. We heard the barrier measures, masking, test, trace, and isolate, testing with providing immediate results. You heard from Dr. Kunowski that more venture cash was raised in 2020 than other years and a lot of important data. And from Priscilla Douglas on leadership. It's all about convening coalitions, consortiums. The effect of COVID on global health patterns was stressed by Professor Lacombe, and Professor Koritskis covered remdesivir, corticosteroids, convalescent plasma, and monoclonal antibodies. Next slide, please. A word of personal conclusion. From the experience I personally had is the need to rethink and re-engineer our clinical trials and their roadmap in order to respond to clinical research questions with fewer ends and a shorter time frame. Next, please. From the New England Journal of Medicine on October 7th, drawing from experience of COVID, Ebola, and HIV, we need multiple strategies for preventing and treating COVID-19, including pre-exposure, post-exposure, and vaccines, since it is highly unlikely that we'll hit a home run on the first trial of any intervention. Next slide, please. The recording of this summit will be available to you all at a short time frame after the summit on YouTube. Please also visit our website, bostonbiotechnologysummit.com. Dear colleagues, I think we shall prevail and everything will be all right at the end. Thank you for attending this summit. It was a privilege setting it up for you. Thank you.